So I think we'll get started now. Just after 11, a few folks be coming in. But so good morning, good afternoon to everyone. Paul Danis, I'm with the consortium, and we're happy to have Caroline Liu with us today to speak about some of her recent work, which is pretty exciting. Let me first give a couple bits of introduction and a couple mentions of some information you may find helpful. First off, we just want to thank all of our supporters who help us to run these kinds of programs and operate the consortium. SEER has been our partner. We also have our major sponsors, Thermo Fisher, Bruker, SCIEX, and Lilly, as well as our supporters, Agilent, Pfizer, and Nuomics, for whom we're all grateful, and we appreciate their support. So if you see them, you can thank them for helping us do these kinds of things. So as most of you know, we have our meeting coming up in less than 2 weeks in Chicago. This will be a pretty exciting meeting. We're going to have a lot of different topics here beyond just mass spec, trying to bridge the genome phenome concept. Proteoforms we think is an important piece of this. We have some tremendous speakers. We have people like David Walt going to speak about non-MS methods to get sensitivity beyond mass spectrometry. Amy Harrod is speaking about some of her exciting single cell work. We have Ing Gerds going to be speaking about some of her clinical applications, which are pretty cool. Lily be speaking about some imaging and spatial work. And then we have Parag and Jeff are going to be talking about non-MS methods, some of these nanopore sequencing and other single molecule sequencing approaches that are pretty exciting. So it should be a great time, a great week, and we hope to see a lot of you there. We're additionally going to have some roundtable discussions where we can all get engaged, because there's still, as you all know, there's a lot of open questions we're trying to address here. The first one, Hartmut be running one, talking about kind of the technology, methodology of proteoform discovery and where that's at. What are the challenges? What are we trying to address there? Next generation sequencing, proteomics, et cetera. Lloyd be running this, where we're going to kind of go beyond mass spec. What are the kind of technologies that are complementary? How's that going to mesh? What do we need to figure out around that? Neil will be running a panel on the Human Proteoform Project, which would be interesting because we also have Doug Shealy, who's with the Common Fund, the NIH. So maybe we'll get some insight on how we fund large-scale projects there. And then Ing's going to be running a roundtable on clinical discussions for proteoforms and how proteoforms would be critical going forward for diagnostics and things like that. So it should be fun. Hope to see you all there. And if you can't make it, we are having an online session for our early career researcher group is running this session on October 3rd, which is open to anyone for no charge. You can go to the website, and if you look under the program, you'll find this, and then you can sign up. It's going to have Kyo Wong's going to be talking about this idea of having a golden data set where we can have a set of data we can all use to sort of test our processing and things like that to see, are we getting the same answers as everyone else, or is there variations on the data processing side? Because we'll have a standard data set we can all sort of test with. Lucas is running a project around MSMS to try to kind of evolve that, which we've had from a couple of years ago, and see what are some standard approaches we can use. Are there some basic on-ramps we can give people? Say, if you want to start with the MSMS top-down, here's a good approach to take based on your technology. And Neil's going to talk in general terms. I'm not sure exactly what, but it's always fun to watch Neil talk. So that'll be open to anyone. If you want to go there, you can sign up by Zoom, and you can join this session here. Otherwise, we hope to see a whole bunch of you there in Chicago, and we should have a fun time. So with that, I'd like to hand it over to Caroline. She's currently with the Department of Chemistry and Biochemistry. She's a research faculty there at Florida State, and she's going to be speaking with us today about advancing top-down protein analysis by tandem-TIMS-MS approaches. So go ahead, Caroline. You go ahead and take over.
Thank you, Paul, for the introduction. So good morning, everyone. First, I would like to thank the consortium for top-down proteomics for this opportunity to present our research and this platform. And yeah, I'm a research faculty in the Blyholder Laboratory at the Florida State. And today I'm going to talk about how we advance the top-down protein analysis by tandem-TIMS-MS developed in our lab. So in our lab, we develop methods to study the primary. I'm sorry, before that, maybe I should laser pointer. Yeah. So in our lab, we develop methods to study the primary, tertiary, and quaternary structure of proteins. And with this, we would like to investigate how protein structures influence the dynamics and the function, especially how different proteoforms can affect the dynamics and functions of proteins. We are especially interested in elucidating the structure of glycoproteins because glycoproteins are involved in many diseases. This is an example. These are 5 spike proteins of 5 different viruses. And so the spike proteins are responsible for the attachment and fusion between virus and the human cell, and therefore they are the target for neutralizing antibodies. All these spike proteins have very similar basic structure. So the protein will build the core, and this protein core are covered by a thick glycan layers, which we call glycan shield. This glycan shield has several functions. First, to stabilize the spike proteins, but also as it helps the escape mechanism. For example, in HIV-1 spike proteins, it can change the glycan profiles of this glycan shield, and this helps the virus to escape the neutralizing antibodies. So this shows how glycoproteoforms influence the dynamics and functions of proteins. Now in our lab, we develop 2 different methods for structural elucidation of proteins. The first one is an experimental method called tandem-trapped ion mobility spectrometry coupled with mass spectrometry. This is developed with collaboration with Bruker Daltonics. And also our lab developed a theoretical or computational methods called structure relaxation approximation, or SRA. And here we obtain the 3-dimensional structure of proteins in the gas phase from the ion mobility spectrum. And so, so far we have published 2 papers on SRA. In the first papers, we elucidate the structure of monomeric proteins, and in the second paper, we study the structure of protein complexes. But in this talk, I'm going to focus on the tandem-TIMS-MS. So first, I would like to give a very brief introduction on trapped ion mobility spectrometry. TIMS cells, so TIMS it consists of the entrance funnel, analyzer tunnel, and exit funnel. And the mobility separation occurs in the analyzer tunnel. So we have a stream of buffer gas that carry the analyte ions into the analyzer tunnel, and the larger analyte ions, or proteins like unfolded proteins, they will have larger cross sections. It means there are more collisions. It experiences more collisions with the buffer gas particles, so that the friction force of these larger proteins is higher than smaller proteins. At the same time, we apply a counteracting electric field, and in such a way that we have a gradient in the first half of the analyzer tunnel and a constant electric field in the second half of the analyzer tunnel. Now, when the sum of the electric field, the force from electric field and the friction force is 0, then the ions will be trapped in a specific location. So larger proteins, because it has also a larger friction force, this requires a higher electric field to offset, to offset the friction force. It means these larger proteins will be held or will be trapped in a different location than smaller analytes or smaller protein, like more compact proteins. And this is how the mobility separations, this is the principle of mobility separation in TIMS. Sorry. In our lab, in the Blahilder laboratory, we developed tandem-trapped ion mobility spectrometry. So here we coupled two TIMS cells, and in the interface we have 3 ion apertures. So far, we have developed 2 tandem-TIMS instruments. The first one is the coaxial tandem-TIMS. We have the TIMS-1 and TIMS-2 coaxially aligned. And in the second instrument, we have the TIMS-1 and TIMS-2 orthogonally aligned. So the first tandem-TIMS paper was published in 2018, and we also have a joint patent with Bruker Daltonics. So now, what are the benefits of tandem-TIMS? Why do we want to use this, or why do we did we develop this instrument? So tandem-TIMS has many components, and in each of these components, there are different operational modes. For example, in each of the ion mobility cells, we can perform mobility analysis, transmission, or we can trap ions for extended period of time. We can also collisionally activate ions. Then in the interface, we can select ions with specific mobility, and we can also perform ion activation. And then later, in the quadrupole and in the collision cell, we can select specific M/Z and also did another ion activation. So by combining these different type of operational modes, we can have a large variety of analysis workflow. And using this different analysis workflow, we can characterize different level of protein structures. Now, in this talk, I will introduce, or I will discuss 3 different topics. The first one, I would like to talk about how we study protein complexes by elevated pressure CID in tandem-TIMS. So now, to study native intact protein complexes, there are 2 established mass spectrometry-based methods. The first one is called collision-induced dissociation. Here, basically, a protein complex ion collide with buffer gas particles, and so it increase the internal energy, and at some point it will eject an unfolded subunits, so unfolded monomeric subunits with extended structure, so unfolded structure. In the second method, surface-induced dissociations, the protein complex collide with the surface, and then it will dissociate the protein complex into subunits. The subunits generated by this method, they reflect the complex assembly pathway, and that's why SID is very useful when we want to study the architecture of protein complexes. But these 2 methods, they have the one characteristic is that they operate at low pressure regime, so lower than 10 to the minus of 3 millibar. But now in ion mobility spectrometry, so all ion mobility spectrometry instrumentations, including TIMS, we are operating in millibar pressure regime. So, for example, this is the pressure, the gas pressures that we are using in our tandem-TIMS. This means that if we want to dissociate ions in the interface between these 2 TIMS cells, we need an ion activations method that also operates in millibar gas pressure regime. So to overcome this challenge, we attach, so here in the interface of TIMS-1 and TIMS-2, we attach metal grids in the electrodes, so in two of the ion apertures. And using these metal grids, we can increase the electric field strength of that are applied in this region up to 1200 volt per centimeters. This is 4 times higher than if we do not use metal grids. And using this high electric field, now we can perform CID at elevated pressure. We want to answer these two questions. So can we perform CID at elevated pressure and produce subunits that are reflective of the complex assembly pathway? So basically similar to a surface-induced dissociation. And the second question is, the subunits that are produced, do they retain the native structure of the protein complex? So as model system, we are using avidin tetramer. So avidin is egg white proteins. It's a glycoproteins with 4 monomeric subunits. And the quaternary structure of avidin is a dimer of dimers. Before we perform the elevated pressure CID in tandem-TIMS, we want to make sure that the precursor ions for CID is the intact native avidin tetramer. So first, we electrospray avidin from ammonium acetate buffer and then perform mobility analysis in TIMS-1. And these are the results. So this is the ion mobility mass spectra. And we can see that the avidin tetramers are dominates the spectrum. And from here, we can obtain, so from the arrival time, we can obtain the cross sections. Now we plot the cross sections of the avidin tetramers as a function of the charge states. And as you can see here, the cross sections obtained by tandem-TIMS, they are very consistent with the RF confining drift tube IMS and also with the structures obtained in X-ray. So this shows that we do retain, so in tandem-TIMS, we do retain the native-like avidin tetramer structure. So now we have this avidin tetramer as precursor ions. We perform CID in the interface of TIMS-1 and TIMS-2 for a variety of activation voltage. And then we perform mobility analysis in TIMS-2 to get the cross-section of the product ions. Now, at activation voltage of 140 volt, we observe compact trimers and also extended monomers. And this means that at this activation voltage, the elevated pressure CID follow this typical CID mechanism. So first, monomeric subunits start to unfold, and then later this monomer will be cleaved, and it will also take up like almost half of the total charge of the tetramer. So this is called the asymmetric charge partitioning. Anyway, this elevated pressure CID at low activation voltage does not really produce the structurally informative dimers. But now when we go to 240 volt, we observe compact dimers and compact monomers. So this is much more interesting for us because we observe the compact dimers. And that's why we start to look at other tetramers like homotetramers, and we want to know if we also see the structurally informative dimers. Now for concanavalin A, this is homotetramer proteins with molecular weight of around 110 kilodalton. We perform CID in tandem-TIMS at 270 volt. And we observe monomers, trimers, but also dimers. Now, when we compare this spectrum with CID performed at low pressure, this CID spectrum only shows monomers, trimers, and truncated tetramers. but they do not produce any dimers. Now, when we compare the spectrum to surface-induced dissociations, they look very similar because here they also produce monomers, trimers, and also dimers. So it means that the tandem-TIMS-CID spectrum of cone A at this activation voltage is very consistent with SID. We observe the same trend for streptavidin tetramers. When we obtain the CID spectrum at 100 volt, we can see monomers, trimers, but also dimers. And these dimers are not observed in the low pressure CID, but they were observed in the surface-induced dissociation spectrum. Now we look at the cross sections of the produced dimers in our instrument and compare them to SID. So this line represents the cross section of cone A dimers in the X-ray from the X-ray structure. And as you can see here, the cross section of the dimers produced by SID are very close to the X-ray structure. At the same time, when we look at the dimers produced by CID in tandem-TIMS, so for the same charge state, so 8, 9, and 10, they are also very close to the cross-section of the X-ray structure. The same thing is observed for streptavidins. So for the same charge state, so 5, 6, and 7, so the dimers with 5, 6, and 7 charges, they have very similar cross-section as the cross-section obtained for the X-ray structure. So this shows that with the elevated pressure in CID in tandem-TIMS, we are able to produce structurally informative subunits from protein complexes. This work has been accepted as publications by JSMS. So this week, the paper is online starting from yesterday. If you're interested, you can read. Please read this paper. And also in the top-down proteomics symposiums next month, I will be presenting a talk about this elevated pressure CID, where I will show more data. Now we are interested also in the nativeness of the subunits produced in, I'm sorry. So we are also interested in the subunits, in the nativeness of the subunits produced by CID in tandem-TIMS. So we look into streptavidin trimer 10+. Here, this trimer are produced by CID at at increasing activation voltage between 60 to 78. And as you can see here, this is the cross-section distribution. So it starts to so the cross-section start to increase with increasing activation voltage. So in fact, if we look at the cross-section distribution at 78 volt, we can fit 4 different Gaussian functions underneath this distribution. Now we apply our theoretical method, the SRA, to predict the ion mobility spectra of the streptavidin trimer 10+ at different ion temperatures. And these temperatures represent the different activations energy. And as you can see here, the cross-section distributions are the cross-section distributions we obtained from SRA, they are very close to the cross-section distributions we obtained from the experimental measurements. So this means that using the structures we obtained from the SRA calculations, we can follow how this trimer unfolds with increasing activations energy. And it also shows that at low activations energy or activation voltage, this trimer still has a compact structure. So with this, I would like to move on to the second topic, which is the study of primary structures using top-down analysis. So for the motivations, I would like to show you this data. So when we look at the pandemic back in 2020, the sequence, the wild type sequence of the SARS-CoV-2 spike proteins has an aspartic acid at the 614 residue. But around February 2020, there is a mutant, like a single mutant, start to emerge in Europe. So it has glycine instead of aspartic acid in 614 at 614 residue. And within a couple of weeks, these new mutants, they dominate, or basically they drove the pandemic. And these observations has been rationalized by the different conformations of the receptor binding domain in the spike proteins. So there are two different conformations for the receptor binding domain: the down conformations, where the spike protein cannot bind to the ACE2 receptor of the human cells, and then the up conformations where it can bind easily to the ACE2 receptor, and this allows the infections, so the attachment of the spike proteins with the human cells. And these single mutants, it seems that these single mutations support the up conformations. So it means that mutations at the single amino acids has a very huge impact on the binding affinity of the spike proteins. And with this, I would like to illustrate how in top-down proteomics, we need 100% sequence coverage in order to catch a mutation at a single amino acid. Now, these are two challenges for top-down analysis by tandem-TIMS to be able to get 100% sequence coverage. It means first, we need to break all the peptide bonds. But however, when you use CID, mostly we can only cleave the peptide bonds in the N and C terminal regions. But in the literature, we found out that UV photodissociations also is able to produce the internal fragments. Then the second challenge is we need an accurate fragment ion assignment, especially because we are working with TOF, so time of flight, which has a mass accuracy of between 10 to 15 ppm. When we look at the random match probability, for example, for carbonic anhydrase, this is a protein with 259 residues. The random match probability, if we consider the internal ions, would be around 40 to 50%, which is very high. So that's why we need to overcome these challenges, we need to integrate UV photodissociations as an ion activations method in tandem-TIMS, and also to increase the accuracy of fragment ion assignment, we developed the MS3 approach instead of MS2, and also we developed our own data analysis software using OMSA scoring function. Now we develop the tandem-TIMS-MS with UVPD capability, starting from a timsTOF Pro instrument. So this timsTOF Pro is commercially available. It has one TIMS cell and then a quadruple ion guide, a quadruple mass analyzer, a collision cell, and time of flight. Then we add a second TIMS cell at the front end. And between these two TIMS analyzers, we added a quadrupolar ion trap. This quadrupolar ion trap operates at a millibar pressure regime. After that, we add an optical setup with a 230 nanometer laser This has a repetition rate of 1 kilohertz and maximum energy of 200 microjoule per pulse. So it means here we can, after ions are eluted from TIMS-1, we can trap ions in the quadrupolar ion trap, and these ions can be irradiated by the laser beam. We also have in this orthogonal tandem TIMS-MS, besides the UVPD, we also have CID, and we are able to perform CID in 3 different locations. Now, as a proof of principle for UVPD, we perform we perform CID and UVPD in protein using protein ubiquitin. When we look at the isotopic pattern for the Y58 8+ ions, we can see a difference in the CID and UVPD spectra. So in the CID spectra, the isotopic pattern they match correctly with the predicted isotopic pattern from the sequence. But when you look into the UV-induced spectrum, we can see a shift of the monoisotopic peak by about 1 or 2 peaks. This shows that there is a loss of 1 to 2 hydrogen atoms in this Y58 8+ ions, and this is expected for UVPD because Y-1 and Y-2 fragment ions are very typical for UVPD. And this finding confirmed that we are able to perform UVPD in tandem TIMS at 2 to 3 millibar pressure. Now we want to develop an MS-3 approach by coupling CID or UVPD with PASEF. So just a short introduction on PASEF. It's an acronym for parallel accumulation serial fragmentation. And this is actually an automated MS-MS analysis. But instead of selecting the precursor ions only by specific M over Z, it also selects the precursor ions by specific M over Z and also specific mobility. And after the selection in the quadrupole, the fragment ions or the ions will be further fragmented in the collision cell. This PASEF method was developed for bottom-up proteomics, and it's very useful. But now we want to use this PASEF in combinations with our CID and UVPD for top-down proteomics. As the first example for coupling CID with PASEF, we use carbonic anhydrase 2. We perform CID at 180 volt between L2 and L3, and we mobility analyze the fragment ions in TIMS-2. These are the ion mobility mass spectra of the fragment ions. So as you can see, there are a lot of different fragment ions. The PASEF algorithm selected 31 precursor ions. So the red dots are the precursor ions selected by the PASEF algorithm. Now I'm showing the PASEF MS-MS spectrum for this particular ions. And so the red lines here show the peak assignment of this MS-MS spectrum. And using this MS-MS spectrum, we can assign this 1267 M over Z to Y67 6+ ions with very high accuracy, because they are, so A is the scoring function, so the probability for random match is very, very low, so 10 to the minus 46. Now we reject all PASEF precursor ion assignment when the minus log A is lower than 15. So from out of the 31 precursor ions, we only accept the assignment of 11 ions. And from these 11 ions are, we have 9 Y-type ions and 2 B-type ions. And also 6 of these fragment ions are related to proline effect. So the cleavage coverage of the carbonic anhydrase achieved by this method was 37%, but I think we achieve a high accuracy and assigning in the peak assignment, because as expected for CID fragmentation of larger proteins, we saw fragmentation in the N and C terminus, and also some due to the proline effect. Now, the second example, for the MS3 approach, we coupled UVPD with PASEF. So at this point, we are using protein ubiquitin. And so we perform mobility analysis in TIMS-1. We are selecting only ubiquitin's charge state 6 here in the interface. So as you can see here, this is the mass spectra of the selected ubiquitin charge state 6 when the UV laser is still off. So you can only see the charge state 6. Now when we turn on the UV laser with energy of 10 microjoule per pulse, around 20 milliseconds. So basically we have only 20 UV pulses. We can see that this UV PD induced fragment ions. Now we limited the PASEF algorithm to select only in this region, and it selects only 2 precursor ions. Now again, we look at the MS spectrum or the PASEF spectrum of these specific ions, and we can assign this 1017 M/Z with very high accuracy as B18-2+ ions. So this shows that with our instrument and our data software, home-built data software, we show that we are able to use the PASEF precursor scheduling and auto-MS for top-down protein analysis, and we have a high confidence in ion assignment using the bottom-up bioinformatics methods. We published this work also in JSMS special issue last month. So this is another paper than the elevated CID pressure, CID. So finally, I would like to show you the structural heterogeneity of the fragment ions produced in tandem-TIMS. So when we first performed CID in tandem-TIMS on protein ubiquitin back in 2018, we observed multiple conformations on almost all fragment ions. So we found this very interesting, and so we look more into it. And so these are the trends that we observe for the YN4+ series of ubiquitin. As you can see here, there are 2 conformational families. So these are more extended, and these are more compact. And within these conformational families, we also see several conformations. Now, so in the fragment ions, we have between 44 to 50 residues. We have a transition from the more extended structure to the more compact structure. This conformational heterogeneity is also, we also observe this on other proteins, for example, cytochrome C, avidin, and also beta-lactoglobulin. So as you can see in this diagram, almost all of these ions, they adopt conformations that are between the globular or compact structure and the alpha helical structure. And most interesting is that if you look at this line, so all these ions here, they have the same mass and also the same charge state, so charge state 4, but they have different conformations. So this means that the cross sections of the fragment ions, they are not they do not only depend on the mass or the charge states, but it's possible that there are other structural informations that are contained in the cross section of the fragment ions. So to study this, to further investigate this observation, we use a different experimental workflow where we so after performing CID, so basically after we produce these fragment ions, we trap these fragment ions in TIMS-2 for extended period of time. So between 0 second to 5 seconds before we perform mobility analysis. And we found quite interesting results. So for the Y42 ions, as you can see here, the cross section of the ions does not really change between 0 second to 5 seconds. So they are all there are no significant change. These ions adopt an extended conformations. Same things for the Y54 plus. So between 0 second to 5 seconds, the ions adopt compact conformations. But now when we look into these ions here, the Y47, at 0 second, most of the ions adopt extended conformations. There are some of them that already have compact conformations. But now when we trap these ions up to 5 seconds, you can see a decrease of these extended conformations and an increase of the compact conformation. We rationalize this finding or these results by using the simplified free energy diagram. So for annealed fragment ions, it has low energy barriers only. So the ions can reach the global energy minimum very fast. So this is shown, for example, for the Y54 plus. So even at 0 second, we already reached the compact structure. But now we also have a metastable fragment ions, which has at least one high energy barriers. So these high energy barriers slow down. So the ions cannot reach the global energy minimum very fast because of the high energy barriers. And this is shown by the Y47 4 plus, which needs to be trapped for 5 seconds before most ions can reach the compact conformations. So these results indicate that most probably the top-down fragment ions can retain some aspects of the native protein structure when they are trapped at 0 second. The structural heterogeneity are also observed in the fragment ions produced by UVPD. For example, this is angiotensin, so it's a nona peptide, and as you can see here, these are the fragment ions, so A6, A6 plus 1, A6 plus 2, and they have different conformations. So A6 has most possibly 2 unresolved conformations, and when one hydrogen atoms are added, then the ratio between these 2 conformations start to change. And when another hydrogen atoms are added into the A6 ions, then the smaller conformations is more pronounced. But to really understand the physical chemistry behind this cross section of top-down fragment ions, we require more investigations, and also we need we also need to do computational approach. So with this, I would like to close my talk. I will skip the summary in interest of time, and I would like to acknowledge and thank the lab members of the Bleiholder laboratory. So Christian Bleiholder is the PI, and also I would like to thank the previous members of our lab, our collaborators from Bruker Daltonics, especially Mark Ridgeway and Melvin Park, and also the funding agencies. And I would like to draw your attention to our annual summer school on tandem-TIMS-MS and PSA and SRA computational methods. So we have we have travel and accommodation stipends for in-person participants. This is funded by NSF. So if you are interested, please follow our website or Twitter account for updates. Also, our lab is recruiting postdocs, graduate, and undergraduate students. So thank you for your attention, and I will be happy to answer any questions that you might have. Thank you.
Great, thank you, Caroline. That's some fascinating work. Really cool stuff. So if you have a question, you can raise your hand, ask it live, or you could type it into the chat, whatever works for you. I just had a quick question on the PASEF. I think it was carbonic anhydrase data where you said you rejected a certain fraction of the ions. Can you talk a little more about your logic and your criteria for rejecting versus including certain ions?
So yeah, I can try. So let me. So I think I have different slides. So this so I didn't show these slides in my talk, but so basically, so this is the log A, minus log A. So A is this scoring function that we use, and as you can see here, so this is the fast discovery rate as a function of this minus log A. So if we have minus log A lower than 15, we start to have a very high fast discovery rate. But if the minus log A is higher than 15, we have very low fast discovery rate. So this is where we draw the limit. So we said, okay, we accept only peak assignment with the minus log A higher than 15. And we say this is accurate enough because the fast discovery rate is very, very low, but we do not accept the peak assignment if they are within this region.
All right, that sounds great. Pretty clear. Thank you. So Moe has a question. He says, very interesting data. Do you see different CCS for the same fragments, but from different charge state precursors?
Sorry. Different charge state precursors. Okay, so I believe this relate to this work. So that's interesting questions. Actually, I believe we have not we do not have any data where we let's say start use precursor ions with different charge states for the same proteins. So usually we start with, let's say, ubiquitin charge state 7 as the precursor ions, and then we look into the fragment ions. But we haven't tried like different charge states as precursor ions. But yeah, that will be very interesting to try actually.
Thanks. So I had another question on your energy curves. Near the end, you were showing those energy curves to rationalize the different conformations. Are these just sort of guesses on the barriers, or do you actually have some computation or something to give you an indication on these barrier heights?
No, this is just like a simplified free energy diagram as an illustration to rationalize why we observe this trend, like these different trends. But we haven't done any really in-depth computational study where we start to calculate the energy barriers for a specific fragment ions. No, not yet. But that will be very interesting to do, and yeah, that will be also like a ongoing work. Yes.
Yeah, that's fascinating. I mean, I don't know the field, but how large an ion can you calculate that for? It seems like you would run out of ability after a while. Can you do some of these larger fragments and calculate these things?
Yeah, so I think Christian will be the person to answer these questions. He's the computational person in our lab. So Christian, if you can be unmute. people?
Okay.
Or
Can you hear me?
yes.
Yeah.
Oh, okay. Thanks. Yeah, that's a very interesting question. So so it depends on the level of theory. So that's what limits the size of the fragment ions that you can calculate. So if you wanted to use I think up to 20 to 30 residues is possible with what's called density functional tight binding methods. They're reasonably accurate. So, but if you want to go to like the Y50 ions here, I think you would have to rely on MD, and I don't really trust that they are really accurate in terms of barriers and so forth.
Okay, sounds good. Be interesting to see how that all works out.
Thank you.
Thanks. Are there any other questions or comments?
Can I have one question here? Yeah, this follow-up question. So if we don't get the energy barrier, at least are we able to get the predict somewhat compact or native-like structure for these ions? Just like a cross-section you would expect for this amino acid sequence, like see how far is it compared with a peptide structure? Because I don't know if just the cross section is just related with the amino acid sequence, or it's just still in the gas phase activated one.
Okay. So yeah, so I'm not sure if I answer your questions correctly, but so what we are going to do in the our next step in this study is so right now we have the cross sections of the fragment ions with specific sequence, but they are like peptides that are produced in situ in the gas phase, right? So now we want to compare the cross sections to peptides, so the same peptides with the same sequence that we produce like from in the solution. So it means we will electrospray these peptides from the solutions. They have the same sequence as the peptides that we produce with the top down, and then we will compare the cross section if they are the same or not. So to get like more understanding about where does this cross section come from? Do they only like you said, if they have these specific conformations or cross section, do they determined by the sequence or not, or by maybe ion activations in the gas phase? Does that answer your questions, Maggie?
Yes, thank you.
Great, thanks. Are there any additional questions or comments people want to throw out? So if not, then thanks, Caroline, for really fascinating work. That's some crazy stuff you guys are doing there. You're going to add 2 or 3 more ion funnels, I assume, in the next few years. So
thank you.
We'll see where that goes. So thank you again. I really appreciate it, and hope to see a lot of you in Chicago. And if you can't make it, don't forget there's that ECR Zoom session on October 3rd you can sign up for if you want to participate at that level. So thanks again. Thanks to our sponsors, and we'll see you all soon. Thank you. Bye-bye.
Thank you, everyone. Bye-bye.